Hello, everyone. My name is Doug Chapnick, and I'm the founder and CEO of a Colorado-based biotech company called BioLoomics. And I'm here today to introduce you to a project we just launched about 2 weeks ago in collaboration with KIF1A.org to apply our protein evolution in a microscope technology to advance KAND therapies. So I thought I'd jump right in and tell you about our project. And then I'll back up and I'll tell you about our company and how the technology works and how it all comes together. But ultimately, our goal with this project is to find candidate treatments for KIF1A. And those could either be small molecules or they could be gene therapies. In the question and answer period, I'll talk about the gene therapies if you want. But this project's divided up into 3 phases. The first phase is where we build an assay to essentially go into chemical space and do high throughput screening to discover a rare chemical that seems to reverse the effects of KIF1A mutants. Once we build that assay, we can actually use the technology in several different ways. And one of those ways is to search for a novel target using a pull down like approach using our technology. And that's in the middle panel here. And so the goal there is to find a novel target within 4 months. And ultimately, within 6 to 12 months, our grand goal is to have a candidate that should go into animal testing, and ultimately, if it's successful there, it would go into clinical testing. And so today I'll just talk to you about the phase 1, which is this building of an assay using our technology to find a high throughput screening assay that doesn't measure wild type KIF1A, but does measure mutant KIF1A. So I'll back up a little bit, tell you about our company. So our company's been around for 2 years, and we're about $1.5 million into this project, the company. And since 2019, we've grown the team to about 6 people, and we've been inventing this evolution in a microscope technology that I'll tell you about. So our corporate mission is to harness the power of evolution to build therapies and have those therapies save lives. And to just give you an example of the power of evolution, we can compare 2 proteins that have evolved. So one is the insulin that's in hagfish, and it's basically an ancestral organism that evolved roughly 300 million years ago compared to the human insulin that we have today. And the difference between these 2 proteins is only 20 amino acids, but the insulin from hagfish is essentially useless therapeutically. And the insulin that courses through our veins is currently used to treat 463 million diabetics in the world, and it drives a $60 billion industry. So it took evolution 300 million years to change 20 amino acids, and then find the exact combination of those 20 amino acids to give ultimately to yield the insulin that we know today as a therapy. And so if we can harness this type of evolution and this selection for very rare sequences of proteins, but we can speed that up and do it in a lab, we have a real shot at building therapies for diseases that lack any therapy. And so that's why I'm here today to tell you about our approach for KAND. So our evolution in a microscope technology shrinks this timeline by using arrays of human cells, where each cell contains a biosensor. And that biosensor enables us to measure some sort of biochemistry inside that cell. And then when we deliver different variant protein designs to each cell, we can ultimately search through designs that affect the biosensor and thus the biochemistry it measures versus ones that don't. And so we can easily converge on designs of let's say, an antibody or insulin that has an increased potential to be a therapy over other members of our variant libraries. So if we dive a little bit deeper, evolution in a microscope really only has 3 components. The first component is synthetic biology, where we build plasmid DNA libraries with the mutations, and we deliver those to biosensing cells. The second component is machine learning and imaging, where we use the microscope to image all 10 million cells that are all arrayed in each 96-well plate in our microscope, and to create plots of the activity inside every single one of those cells and how every variant protein design that we've introduced to those cells affects biochemistry we're monitoring. And then lastly, once we know where the rare variant exists inside that plate, so we have the XY coordinates of something we want to, a cell that we want to retrieve, we've built a robotic cell sorter, essentially a cell vacuuming device that's attached to the microscope that then goes and vacuums this cell that is has the winning design so that we can extract the sequence. And the real motivation for applying our technology in the KAND space is that we want to evolve drug discovery assays that are suitable for high throughput screening. Really, most assays out there that are off the shelf are more for one-off experiments in R&D. And in the high throughput screening community, we have a metric to know what's the difference between a great assay, a good assay, and a bad assay in high throughput screening. And we use this metric that's displayed here on the screen, an equation for the Z prime or the Z factor. And what it boils down to is, if you can fit a lot of standard deviations into your dynamic range, you have a good assay. And if you can't, you have a bad assay. And what a bad assay is you have a lot of false positives. You'll have drugs that you think are candidates and they won't pan out downstream. And so we're on the search for a good assay, a high Z prime assay for mutant KIF1A protein expression. And so I'll just dive into a little bit of how we develop our biosensors, and those are the assays we're developing here in this project. And so I'll tell you about an example of where we use evolution in a microscope to build an ERK kinase biosensor. So ERK kinase is a commonly used effector in multiple pathways, mostly in oncology and wound healing. And the existing assays that were out there that we surveyed didn't have a Z factor that was high enough for us to kick off one of our pipelines that's not related to KAND. But I'm going to tell you about how we built that biosensor, and then we'll transition to how we're going to do almost the same thing to find a KIF1A mutant biosensor. So each one of our biosensors is a long polypeptide chain that's expressed inside cells that has multiple modules or domains that have different function that contribute to the biosensing function of that protein. So these are FRET biosensors. They encode a YFP and a CFP at their N and C terminus. And when those fluorophores, so YFP is yellow fluorescent protein, CFP is cyan fluorescent protein, and they're derived from jellyfish, and they basically make the cells fluorescent with two different colors. And when those N and C terminus fluorophores come together, a property called Forrester resonance energy transfer occurs. And as those fluorophores separate in space and in orientation, the FRET goes down. So the trick to building a biosensor is to somehow couple the distance between the CFP and the YFP to some sort of biochemistry in the cell. And this is an example here of a FRET biosensor for kinase activities. So this is actually the existing ERK biosensor that we then mutated to make a new one that's high throughput screening capable. And so this biosensor doesn't just have the two fluorophores, it has a domain that's a phosphobinding domain called an FHA domain. And that domain folds and binds to a phosphorylated peptide with a threonine phosphorylation. So there's four components to these biosensors. There's the fluorophores, there's two components there. There's the phosphobinding domain, and then there's a substrate domain that gets phosphorylated to the small peptide that's inside the protein. And when a kinase phosphorylates that domain, you get a hairpinning of the biosensor and ultimately a dramatic increase in FRET. And so the trick to making a new biosensor is to mutate that phosphorylation domain, that phosphopeptide sequence, until you find a substrate that's good for another kinase. And the advantages of these FRET biosensors is that there's multiple advantages. One is that they're ratiometric. And so if you picture a cell and you stretch it out, the fluorescence of a fluorescent cell will go down. And so that's an artifact generated from cell shape. And these ratiometric FRET biosensors that we're building, they don't have those artifacts, because as you change the shape of a cell, the CFP and the YFP will change proportionally. So this ratiometric nature uncouples cell shape from biosensor activity. And so that's valuable to us. it translates into lower off-target, lower false positives. Another advantage is that they're non-destructive, meaning that we shine light, we get the fluorescence out, and the cell remains alive. We don't rip it apart. And so what that means is we can shrink our experiments down, which increases our throughput. And ultimately, it also means that we can sample those cells over and over again. And that means more data. And the last one is pretty simple. Because we deliver these biosensors to cells, they're genetically encoded, and we do that once, and they're stably transgenes in those cells. We don't have this labeling prep step that other assays have, which like where you label cells with dyes and maybe you wash the dye out. So essentially, we've deleted a whole step that's pretty normal for high throughput screening by doing this genetically encoded biosensor. So when we have a good FRET biosensor, we can screen up to 100,000 drugs a day. So the way we do that is we seed these cells that contain the biosensors into each well of 1536 well plates, and ultimately every 10 minutes we can cycle those plates, and it's only a 10-hour day to 100,000 drugs. And so what's displayed here in the middle is a picture of cells that this is actually the ERK biosensor that I'll show you the final results of, where without stimulation, the FRET activity is low and it's fairly uniform. And with stimulation, so we added EGF to stimulate EGFR, which ultimately leads to ERK phosphorylation and increase in ERK kinase activity. And so you see a dramatic increase in almost every single cell with our new modified FRET biosensor. And the data is also fairly clean for population statistics. So you can do single cell or you can do population statistics. And here on the right is a graph of minus plus EGF stimulation. And you can see halfway through the time course, we stimulate EGFR with EGF, and you see a dramatic increase in the FRET activity of the biosensors. And there's a fairly low error for each time point. So our synthetic approach to evolve an ERK biosensor, which was our very first proof of concept, was to build an oncology drug screening tool that we could then use for one of our pipelines, was to mutate the surrounding amino acids around the threonine inside our prototype FRET biosensor, and then screen millions to billions of these biosensors in batches, where every cell has one of them. And we're searching for the cell that has the rare feature of when we add EGF here on the right, we see stimulation. And so we're basically saying we know what the graph should look like. We need to search for the cell that will yield that graph. So inside our microscope is an array of 10 million cells. And so a real challenge building our technology was to be able to track every single cell, not just what the biosensors are doing, but the cells are moving. And so we have to physically track them in the XY plane and link their biosensor activity from one time point to the next. So we do that in several different stages. So from left to right here, we label the nuclei to get the center of every single cell. And so we just use a simple DNA dye. These are all live cells. The DNA dye just leaks into the cell. And so we use machine learning to find the center of every single cell using the image of the nuclei. And we use a mixture of machine learning and heuristics to find the outline of every single cell, even though these cells are extremely well packed in those Petri dishes. So they're essentially a monolayer of cells where there's no space with no cells. And so using that cell segmentation tool that we've built, we then apply the outlines of every cell to other fluorescent channels. So we apply it to our CFP channel and our FRET channel. And with those two pictures, we can calculate the FRET ratio for every single cell at every single time point. But each cell moves during the experiment. So these live cell experiments, the microscope is essentially an incubator. And so we're also using the centroids of every cell, the center of every cell, to track every cell and link the region of interest and the biosensor activity from each cell from one time point to the next, ultimately outputting what you see here on the right, which is a heat map where every line is a cell. And we can pinpoint exactly which cells appear to have biosensor fret activity change when we add our stimulus. So in this case, it was EGF. And so outlined in yellow are all the variants in the library that had some sort of EGF response. And using these plots, we can predict whether we could have a really high Z prime score or a low Z prime score. And ultimately in validation, we'll hash that out. So like I was telling you before, there's a third component to this pipeline where we have to retrieve the cells that have these rare variants of gene designs, in this case of biosensor designs. So we built a proprietary cell picking hardware. It's like a tattoo machine in reverse, and it controls the suction of a needle that's essentially the size of a human hair. And so it can vacuum one cell or it can vacuum clumps of cells. And in the middle panel here, I'm showing you an example of a monolayer of fluorescent cells that we then use the cell sorter, the cell picker, to go and extract various different patterns of cells from the plate. And then we measured whether those cells remained alive or dead a day or 2 days later. And so right now, our average, after lots of R&D of using our cell picking hardware, is that we kill about 8% of the cells and leave 92% of them alive and healthy. And so that's pretty good. We can work with that and do our entire pipeline to find biosensors in our libraries. So once we pick cells, there's several contaminating cells that could be picked with the target cell. And so we do a sort of follow-up experiment where we spread the cells out and grow them into discrete colonies that don't touch each other. And then we can re-pick, basically purify the variant away from any contaminating variants in 2 steps. So this is an example of where before picking, there's several colonies and we targeted one, and then after picking, we extracted the colony we were after, and then it's in the needle and we're taking a picture of it to show that we're ready to transfer that to whatever well or petri dish we're interested in transferring it to. And so this cell picking is one of the key aspects of our evolution in a microscope platform, because it mimics natural selection. Basically, all the rest of the cells are dead to us in the same way as organisms die in a real evolution, because our picker is what selects for survival. So this is the output. So we built a better ERK biosensor than was already available. So there was what's called the state-of-the-art ERK biosensor out there. There's about 100 publications. And so we're after one that's high throughput screening ready, because the state-of-the-art one that's out there is not. And so its Z prime is displayed here on the left. So there's some pictures where it's minus plus EGF. You can see there's a sort of muted response. And that's a pretty good biosensor for the ones that are out there. I've personally made several in the literature, and I'm very excited about what we're doing and how we're sort of selecting for only the best assays in our corporate mission. And so the Z prime was negative, so it's essentially not suitable for high throughput screening, although it actually produces fairly decent data for literature-based low throughput studies in R&D. And so that's why it's been in 100 papers, because that's the type of studies that have been using this tool. But ultimately, for what we're trying to do with this pipeline is we're trying to build a new oncology drug, and we're going to need to screen through the hundreds of thousands to millions of drugs to find a rare drug. And so we need that Z prime value to be much higher than what the state-of-the-art ERK biosensor was. And so the one that we outputted is here on the right. So our evolution in a microscope technology selected for a rare variant that measures ERK activity that we found by treating cells with EGF, and the data is displayed on the bottom right, and the Z prime value was 0.6. So we're ready to kick off that pipeline, and we actually did it 2 weeks ago. So this was our first real big proof of concept that we can use our technology to potentially go out into our libraries and search for a biosensor that measures almost any biochemical pathway. And so this evolution in a microscope technology is really about taking libraries of genes and sorting through them to find rare features in a variant. And so that isn't just limited to cell assays and looking for biosensing proteins. We can also do the same thing by using those biosensing cells to go search for smart protein drugs. We're on the hunt for biosensing nanobody drugs right now. We could also search for canonical protein drugs, like small antibody drugs. And then we could also put these types of small nanobody drugs inside cells and monitor when we see disease-like states go away in our cell model systems, and then pull those antibodies out and find out what's bound to them. And that's a sort of strategy that we're hoping to deploy here for this KAND project. But the idea is that what comes out of that pipeline is the name of a potential target that we didn't know about. And then because that nanobody was found by neutralizing the effects of essentially the disease state in our model system, what it bound seemed to cure the disease in the model system, the nanobody has a potential to be a gene therapy. And so we say that sort of the fifth application of evolution in a microscope is also to produce gene therapies. And so our tech is fairly well equipped to make an impact for KAND therapies. And one of the reasons is it doesn't seem like there's a high throughput assay that's very specific to KIF1A mutants over wild type. And so we can go after that. Another thing is that there's limited mechanistic understanding to KAND, and we have a sort of unbiased, throw everything at the wall and pull the rare variant out that seems to work approach. And that doesn't really require any mechanistic understanding besides the name KIF1A and what the mutations that we're trying to select for are. And so another aspect that I was just telling you about how we can fish for drug targets using this sort of nanobody capture approach is that there's no real drug targets been proposed beyond KIF1A that we're aware of. So our tech can basically build high throughput assays, so it satisfies the first feature of no high throughput assays. It doesn't require the mechanistic understanding and has potential to identify drug targets. So we could potentially make an impact in all three of these areas over time. So our synthetic biology approach is to repeat everything I just told you about the ERK biosensor and how we built that. We have libraries already of these biosensors. So to date, we have 10 to 20 million of these biosensors made. They're both in 293 T cells, and we're transferring them right now into SKNBE2 neuroblastoma cells, specifically for this KIF1A project. So we've already made all the biosensors. We just have to find the rare variant that does what we're after. And if you look down to the right, essentially the same idea as what we used for the EGF stimulation, we're going to use here to capture KIF1A mutant biosensors. So biosensors that measure a kinase downstream of the mutant's effectors. And the way we're going to capture them and identify where they are in our libraries is by programming every cell to express KIF1A in a doxycycline inducible system. So we'll add doxycycline. Every cell will make a mutant KIF1A and we'll look for the biosensor that changes. We'll extract that and retest it for wild type KIF1A and ultimately filter down to find biosensors that are very specific for mutants over wild type KIF1A. So the current status of this project is we're pretty much 100%, by the time this airs, we'll be 100% complete with our biosensor cell library construction. So we have 2 cell lines. We'll have 10 million different biosensors in each, and we'll be in the process of introducing our inducible KIF1A genes through this doxycycline inducible system to those biosensor cell libraries. Once that has been achieved, we'll be able to perform experiments that we perform weekly here at BioLoomics, where we conduct searches through libraries using our microscope, ultimately to select a rare cell and sequence the gene inside that cell to, in this case, identify a biosensor that responds to KIF1A mutants, but not the wild type. And so the last step is to confirm that we have that specificity of mutant KIF1A over the wild type. So once we have all that, we have an assay that can then be used to go screen for drugs that appear to neutralize the effect of KIF1A mutants, but has no effect on the wild type. So let me tell you a little bit about how you can help this project. So there's a lot of different things that people can do to help us be successful and try to find a therapy for KAND. You could donate to KIF1A.org and enable us to grow the team for the project, and also for KIF1A to get out there and do what they do. You could also partner with us to screen small molecules. So if you have small molecule libraries and you're interested, once we have our assay, in searching through those libraries, then contact me at bioloomics.com. In a similar way, if you're interested in confirming our assay, maybe perhaps you have a lower throughput assay or even a high throughput assay that we're not aware of, we would like to confirm what we're seeing is in line with what is being studied in other people's labs. So if you're interested in that, please do the same. Contact me at bioloomics.com, particularly if you're interested in comparing your mouse model to what is being found with our cell culture model system. Another thing that you could do is to follow us and spread the word by sharing our mission via social media. We're on LinkedIn, Facebook, and Instagram. And I have a special thanks to the organizers for inviting me to represent BioLoomics and for doing the partnership with us in the first place. It's a very exciting time for us. This is one of the first real tests of our platform, and it's a pretty fairly real world test. So I hope to next time touch base with the KIF1A or the KAND conference and tell you about the success that we have achieved. So thank you for your time, and I'll take questions. Thank you.